So tonight we're going to go over Chapter 13. And in Chapter 13, we look at how genes give us the traits that we have and how the individual traits are passed from parents to their offspring. So Gregor Mendel is recognized as the founder of genetics. And this is the science of genes, heredity, and also variation in living organisms. So he was an Austrian monk who lived in the mid-1800s, and he carefully documented how certain traits in pea plants are inherited. So he bred and kept meticulous records of the results of over 29,000 crosses of the plants, looking at individual characteristics. And also he described how those characteristics were inherited using mathematics. He also studied how multiple traits were inherited together. So many of the theories that he proposed from his research of how genetics works were later actually shown to be true, and they're the basis of all modern genetics. So a popular theory in the 1800s was blending inheritance. And so this means that offspring received a smooth blend of the traits from their parents. So just like mixing coffee with creamer, this meant that the offspring should be a continuous blend of the parents' traits. So if this were true, if you crossed a white rose and a red rose, what would you expect to get? Exactly. So blending inheritance is kind of in between both the parents. And so it's just watering down of both the parents' genetics and somewhere in between. So Mendel started with garden pea plants that bred true for a particular trait in his studies. And so breeding true means that the trait stayed the same in all descendants generation after generation. So, for instance, in this picture you see if you cross a red and a red, you're always going to get a red flower. And that means that those two parents breed true for that red trait. So this also means that if you self-fertilize a red rose, that you also get the red rose as the offspring. So when Mendel cross-fertilized pea plants with different traits, such as different flower colors, the traits of the offspring appeared in predictable mathematical patterns. And so he concluded that hereditary information is passed from one generation to the next in discrete units. And what we now know as genes in the DNA sequence are these units that he was describing. And so through his studies, he realized the theory of blending inheritance wasn't true. So if you cross a red flower with a white flower, in general, you're not going to get a pink flower. So the plant Mendel used in the studies was a garden pea. So this plant has many distinct characteristics that vary among plants. And so some of these characteristics include plant height, flower color, pea characteristics, and pea pod characteristics. And so he studied all these different characteristics between the various pea plants that he bred. So the pea plant is naturally self-fertilizing, and it has an anther that makes male gamete and a carpel that makes female gamete. And both of these are in the same flower. So Mendel controlled the breeding between the pea plants by removing the anther from one plant and brushing the pollen containing male gametes onto the carpels of other plants. So this way the plant would not self-fertilize, and you could specifically breed one plant with another. So today we know that individuals of a species share certain traits because their chromosomes carry the same gene. So the DNA sequence of each gene occurs at a specific location, or what's called a locus, on a particular chromosome. So for instance, all humans carry the gene for skin pigmentation on chromosome 15, and the gene for an LDL receptor, so this is the bad fat, or bad cholesterol, and this is what's involved with heart disease, is at a specific location on chromosome 19. So again, locus of a gene is a specific location on a particular chromosome. And so this picture shows just various genes that you might recognize and what chromosomes they're located on. So as we know from last week, diploid cells have a pair of genes on pairs of homologous chromosomes. So these genes are found at the same location on each homologous chromosome at the loci for that gene. So the two genes found on a pair of homologous chromosomes may be identical, or the alleles may be different. And you can see this in these pictures. So, if the alleles for the gene are identically the same, and so they have exactly the same DNA sequence, the organism is said to be homozygous for that gene. If the alleles are different and one chromosome has an allele that has a slightly different genetic sequence or DNA sequence, because of maybe a mutation, so there's a mutation on one and the other one doesn't have a mutation, then that organism is said to be heterozygous. So in the pictures, the homozygous organism can have either two of the capital P alleles or two of the lowercase p alleles. So those two top pictures up here. So these two up here are homozygous because they have two capital P or two lowercase p. And if they have different alleles, then they're termed heterozygous. And in the picture, you can see this with the one capital P and one lowercase p. So having different alleles may result in different versions of the trait. And the variation of alleles is the basis of all the differences between them. So the particular set of alleles that an individual carries is called the genotype. So for instance, in a pea plant, the gene that codes for a flower color can be either homozygous with two capital P's or two lowercase p's, or it can be heterozygous with one capital P and one lowercase p. And the specific alleles for the flower color that the chromosomes carry is the genotype for that gene. Another term you need to know concerns the observable trait. So this is what you see. So the observable traits, such as whether the flowers in the pea plants are purple or white, are called the phenotype. So genotype is the actual gene, phenotype is what you see. So the genotype directly determines what the phenotype for that trait will be, but it's much easier to see an organism's phenotype than it is to know the underlying genotype that causes those characteristics. So an allele is considered dominant when its effect masks that of a recessive allele paired with it. So a dominant allele is indicated by an italic capital letter, such as the capital P for purple flower color in a pea plant. A recessive allele is indicated by an italic lowercase letter. So in this case, it's a lowercase p for the white flower color in the pea plant. You can tell that the recessive allele is recessive when the phenotype of the heterozygote, such as the purple pea plant in the middle, is the same as the homozygous dominant, such as the purple plant on the left. So the homozygous recessive pea plant for flower color has white flowers, and you see this one right here on the right. So essentially what you're saying is this dominant capital P for purple covers up the white lowercase p, which gives a white characteristic trait. So this is a dominant allele, the capital P, and the lowercase p is the recessive allele that's being covered up. So when Mendel crossed plants, he crossed plants that bred true for purple flowers with plants that bred true for white flowers. And when he did this, all the offspring had purple flowers. So Mendel did not really know why this pattern occurred, because remember, according to the theory at the time, you would expect some kind of light purple flower color. So we now know that flower color in the pea plants is governed by one gene, and the allele that codes for purple flowers, or capital P, is dominant to the allele that codes for white flowers, or lowercase p. Remember, he didn't know this at the time. He did these experiments that pretty much determined how this genetics works. So a homozygous pea plant with two capital P alleles has purple flowers, and one with two lowercase p alleles has white flowers. And remember, the heterozygote with one capital P and one lowercase p also has purple flowers because the dominant purple allele masks the white recessive allele. So this slide looks at the laws of segregation. And so when homologous chromosomes separate during meiosis, the gene pairs on those chromosomes separate also. So each gamete that forms carries only one gene of a pair, and homologous dominant plants make only gametes that carry the dominant allele that are seen in the picture. So if you look at this, you start out with homozygous dominant, and therefore when you get to the next step, you have DNA replication, and the four gametes that are made from that original parent is going to be four dominant alleles in the gamete. So each gamete that forms carries only one gene of the pair. So a homozygous dominant plant makes only gametes that carry the dominant allele. Homozygous recessive plants make only gametes that carry recessive alleles. And a heterozygous plant makes equal numbers of gametes that carry either the dominant allele or the recessive allele. So if you had a heterozygote, so one capital P and one lowercase p, you're going to have four gametes that form, and two of them are going to be capital Q, or dominant alleles, and two of them are going to be recessive. So when homozygous dominant and homozygous recessive plants are crossed, only one outcome is possible. So a gamete with a dominant P allele meets a gamete with a recessive P allele, and so all that first generation of offspring are going to be heterozygote. So one gamete from the parent on the left fuses with a gamete from the parent on the right. So the genotype is capital P, lowercase p. So again, this right here is the genotype, and the phenotype is going to be purple. So the picture shows gene segregation that we learned about last week. So homologous chromosomes separate during meiosis. So the pairs of genes that they carry are going to be separated too. So each of the resulting gametes carries only one of the two members of each gene pair. So a Punnett square is a diagram used to predict the genetic and phenotypic outcome of a cross. So when you're only looking at one gene, it's pretty easy to keep things straight. But once you get started with two or more genes that you're studying, Punnett squares are necessary to keep straight the combinations that are made when the gametes come together. So to use a Punnett square, you first want to figure out the gametes that are produced by the parents and write them on the outside of the square. So in this example, we put the recessive gametes that are produced by the homozygous recessive female on top. So this one would go right here. And so we have recessive gametes. And then this is over here. So these are the dominant gametes. And then a Punnett square to fill out is real easy. You're just going to cross the two. So in this first square, and so each of these are going to end up being the same. There's only one result that comes out of this, crossing homozygous dominant with homozygous recessive. You're going to get heterozygotes in all four. So breeding experiments use test crosses to determine whether the tested individual is heterozygous or homozygous. So since you can't tell whether an organism that expresses the dominant phenotype, such as purple flowers, is homozygous dominant or heterozygous, you do a test cross to determine the genotype. So in order to do a test cross, you cross the individual that has the unknown genotype with the one that's homozygous recessive. So, for instance, in this slide, you see the phenotype is purple flowers. But purple flowers can mean two different genotypes, either homozygous dominant or heterozygous. Either of those would give purple flowers, and you don't know which one it is. So in order to find out, you can do a test cross. So in the example of a pea plant with purple flowers, again, the plant can be either one. So if you cross the unknown purple flowered plant with a plant that has white flowers, so remember, if it has white flowers, you know what it is because it can only be homozygous recessive. So if you cross this unknown genotype that has purple flowers with a known genotype that has white flowers, so it's homozygous recessive, and you get all purple flowers, what do you think this original genotype is? Is it homozygous dominant or heterozygous? Let's go ahead and put we'll go ahead and put the the test cross, the white ones in here. So homozygous recessive up top. So therefore, let's just see what we have. So the unknown, if it were homozygous dominant, let's see what we end up getting. Remember, if we cross each of these, we're going to get 4 heterozygous. So all 4 offspring are going to be purple. And so therefore, if we get 4 purple plants, we know that that unknown was homozygous dominant. If, on the other hand, you cross an unknown with the known homozygous recessive and you get any white plants, you know that this instead, this unknown, was a heterozygote. So let's just look at that. So let's put the known up here, the white flowers that we know, and let's see what happens when we cross it with a heterozygote. So when this happens, we're going to get half the flowers are purple, so half of the offspring are heterozygotes, and half of them are going to be homozygous recessive or white. So again, with the test cross, what you're doing is you're crossing it with the one known that you know, which is a homozygous recessive or white flowers. So you can tell what the genotype is by the phenotype. And so you cross that with this unknown purple flower, and from that you can determine what the genotype is. That's the purpose of a test cross. So another breeding experiment that was done is called a monohybrid cross. And so this checks the dominance relationship for a single trait. So at the time he didn't know, whether or not purple is actually dominant to white or how that works, or and so he did these experiments to see how this works, if purple is dominant to white or how it works. So, so a monohybrid cross checks this dominant relationship for a single trait. So monohybrid cross is a breeding experiment in which 2 individuals that are heterozygous for one gene are crossed. So the frequency of traits among the offspring offers information about the dominance relationship between the alleles. So in this experiment, a plant that breeds true for purple flowers, which is a homozygous dominant plant, and a plant that breeds true for white flowers are crossed to get all heterozygote offspring. You can see that first. So that's the first step. So we make sure all the offspring are heterozygote. So this is the first generation right here. And then you take these hybrid plants, meaning they're all heterozygotes, and you cross 2 heterozygote plants. So the 2 types of gametes from each plant can meet in 4 different possible ways. And it's shown in this Punnett square of how that works. So the dominant sperm and egg can meet forming a zygote with a homozygous dominant genotype. So that's what you see in this top left-hand corner. You can also have a dominant sperm meeting a recessive egg or a recessive sperm, meaning a dominant egg, and both of these form a zygote with a heterozygous genotype. And so that you're going to see in both of these. And then finally, you can have a recessive sperm meet the recessive egg, and this forms a homozygous recessive zygote. So the probability that the second generation of offspring have purple flowers is 3 purple to 1 white. So the phenotypic ratio of this cross is 3 to 1. So Mendel studied 7 individual traits. So he looked at seed shape, seed color, pod texture, pod color, the flower position, stem length, and he found that all these traits of the pea plants were inherited in the same pattern. So only the dominant phenotype in the F1 generation, so all these plants had that, what we just looked at, and there were only dominant phenotypes when you cross two true breeding type plants. And then also there was this 3 to 1 ratio of dominant to recessive phenotype in the F2 generation. So the 3 to 1 phenotypic ratios in the F2 offspring of a monohybrid cross became the basis of Mendel's law of segregation. So this is the ratio that's characteristic of any single trait whose inheritance pattern follows what's called Mendelian genetics. So Mendel's law of segregation says that the 2 members of each pair of genes on a homologous chromosome will end up in different gametes during meiosis. So this idea that you start out with 2 homologous chromosomes and those homologous chromosomes end up, the different genes go into the different gametes is the law of segregation. So a monohybrid cross allows you to track alleles of 1 gene. So what about if you're studying 2 genes? So as we learned last week, when homologous chromosomes separate during meiosis, either one of the pair can end up in a particular nucleus. And this happens independently of the other chromosome, depending on how the chromosomes line up at metaphase 1. So therefore, gene pairs on one chromosome get sorted into gametes independently of the gene pairs that are on other chromosomes. So this is Mendel's second law, or law of independent assortment. So Punnett squares can be used to predict inheritance patterns of 2 or more genes simultaneously, such as in a dihybrid cross. It only does this if the genes actually are assorted independently. And I'll get to the specifics on that in a second. So in Mendel's experiment, he started with 1 parent plant that breeds true for purple flowers and tall stems. So it has a genotype capital PPTT. And he also used a plant that breeds true for white flowers and short stems. So the genotype of this plant is lowercase PPTT. Sorry, I'll skip to the next one. So each plant in this cross makes only 1 type of gamete. So the gametes either have capital PT or they have lowercase pt. And so all the offspring, or F1 offspring, are going to be dihybrids or heterozygotes for both of the traits that are being studied. And so they all have capital T, lowercase p, capital T, lowercase t, and they have purple flowers and tall stems. So in the next step, he crossed 2 of these dihybrid plants in the F1 generation. So in order to figure out the gametes, we use a Punnett square. So look at each parent and mix the first allele with each of the alleles of the second gene, and then mix the second allele with each of the alleles of the second gene. So if you look, you go like that. So cross that capital P with the capital P, and then do the same with the lowercase p. Or, sorry, do the same with the capital T. Sorry, let's start over. So let's start with this one on the left here, and we're going to go. We're going to go like this. So capital P with the capital T, capital P with the lowercase t, and then you go to the next one and do the same. So going across capital P, capital T, then we take the capital T, lowercase t, and then we move to the next one right here. And so that's going to be the lowercase p with the capital T and the lowercase p with the second one, the lowercase t. So that's how you do the gamete. So let's start again. Let's do instead, just to practice doing the gamete, so let's do capital P, uppercase T, lowercase t. So the gametes that are produced, you're going to start with right here, and so you're going to produce a capital P, capital T. So then you go to the capital P and grab this lowercase t, the capital P, lowercase t, then move to the next one, so right here, the other capital P, and it's going to be the same thing, capital P, uppercase T, capital P, lowercase t. Those are the 4 gametes that are produced. So again, let's do the next one over here. So this is the father. Let's see what gametes are produced by him. It's going to be the same as the mother. So capital P, capital T, capital P, lowercase t, and then the next one, lowercase p, capital T, and lowercase p, lowercase t. And if you cross these sets of gametes, this is what you end up getting. And if you look, what's interesting, if you're just looking at a single trait, so let's just look at the flower color because that's most obvious, and you're going to get 1/4 white and 3/4 purple. So looking at one trait, you're always going to get this 3-to-1 phenotypic ratio with a hybrid cross. So, but also something else you get is a different ratio with the phenotypes. You end up getting a 9-to-3 to 3-to-1 ratio looking at both of the traits. So you have 9 tall and purple flowers. You have 3 short with purple flowers, 3 tall with white flowers, and 1 short with white flowers. So again, if you're looking at one trait, you're going to, with Mendelian genetics, you get a 3-to-1 phenotypic ratio. If you're looking at 2 traits in a dihybrid cross, you get this characteristic 9-to-3 to 3-to-1 phenotypic ratio. And so Mendel discovered this 9-to-3 to 3-to-1 ratio in all of his dihybrid experiments. And he noted that each of the individual traits still kept that 3-to-1 ratio that he discovered earlier, even though he was looking at 2 different traits. So individually they have 3-to-1 ratio. Looking at both traits together, you have this 9-to-3 to 3-to-1 ratio. So from this experiment, he concluded that each trait or gene pair sorted gametes independently of the other trait. And this made up that what we just saw of the independent assortment. So not all of the traits that Mendel studied are coded by genes on different chromosomes, even though his results show that all the genes he studied were sorted independently of each other in the gamete. So genes that are far apart on a chromosome tend to assort into gametes independently, even though they're on the same chromosome, because crossing over occurs between them very frequently. So on the other hand, genes that are very closely together on a chromosome are termed linked. So genes that are really close to each other don't assort independently because the crossing over event doesn't really happen all that often between the 2 genes. So if you look at these 2 genes, A and B, right here, in order for the genes to assort independently of each other, you have to have this crossing over event between them. So the further the genes are away on these chromosomes, so let's say the genes instead are on opposite ends of the chromosome, you're more likely to have a crossing over event happen between those genes and have those genes assort independently. So the closer the genes are together, the more likely those genes are to sort together between the cells, or into the gamete. So Mendel studied inheritance patterns are generally examples of simple dominance. So in this case, each dominant allele fully masks the expression of a recessive one. So, but there are other patterns of inheritance that are not so simple. So you can have codominance, incomplete dominance, epistasis, and pleiotropy. So with codominant alleles are both expressed at the same time in the heterozygote. And so one doesn't actually mask the expression of the other. So this can occur in multiple allele systems where there are 3 or more alleles, and this is the underlying system of ABO blood typing. So your blood type is determined by which 2 of the 3 alleles of the ABO gene you have. So the A and B alleles are codominant when they're paired. So for instance, genotype AB gives a blood type of AB, whereas the O allele is recessive. And so it's masked when it's paired with either A or B. So either genotype AA or AO gives a blood type A. Either genotype DB or BO gives a blood type of B. And a blood type of O is only found with the genotype OO. So, and then if you have AB, again, genotype AB gives the phenotype AB. So incomplete dominance, the heterozygous phenotype is in between the two homozygous phenotypes. This is similar to the blending inheritance theory that we discussed at the beginning of the lecture that kind of showed in general not to be true. But in some situations, it does actually happen this way. So incomplete dominance is a condition in which one allele is not fully dominant over the other. So the heterozygous phenotype is between the two homozygous phenotypes. So in snapdragons, one allele encodes an enzyme that makes a red pigment. And the allele, the recessive allele doesn't make any pigment. So therefore, homozygous dominant genotype gives a red phenotype, the heterozygous genotype gives a pink phenotype, and the homozygous recessive genotype gives a white phenotype. And so a cross between a red and white snapdragon yields a pink one, and the cross between two pink snapdragons, so this is the heterozygote, yields a 1 to 2 to 1 ratio of red, pink, and white. So what happens normally in this incomplete dominance is in, for instance, in this, the dominant gene encodes for red, and the white gene, the one that gives this white color, codes for defective enzyme. And so therefore, usually when this happens, you have just less of the enzyme coding for red, and so you're going to get a less intense color, which gives the pink. So some traits are affected by multiple gene products. And this effect is called polygenic inheritance, or epistasis. So one example of this is the Labrador Retriever coat color. And so you can have 3 different coat colors, black, brown, or yellow. So in Labrador Retrievers, a dominant allele, B, specifies the black fur, and its recessive partner, lowercase b, specifies the brown fur. So a dominant allele of a different gene, E, causes color to be deposited in the fur, whereas the recessive partner, or the lowercase e, reduces the color. So a dog with both dominant genes has black fur. A dog with a dominant E allele, which is either homozygous or heterozygous for that E allele, and it's homozygous recessive for the B or brown allele, ends up being a brown dog. Whereas the dog homozygous recessive for the E allele has yellow fur regardless of the alleles for the color, the coat color. So because if it's homozygous recessive for that E allele, it's not actually depositing any color into the fur. So this is an example where you have 2 different genes that affect the coat color, and they depend on each other. So a pleiotropic gene influences multiple traits. So this is instead of 2 genes affecting one trait, you have 1 gene affecting multiple traits. So mutations in pleiotropic genes are associated with complex genetic disorders. So different diseases such as sickle cell anemia, cystic fibrosis, and Marfan syndrome are all examples of things that are caused by 1 defect in a gene. So for instance, in Marfan syndrome, this is an example of a mutation caused by, or a disease caused by a mutation in a pleiotropic gene. So mutations affect the elasticity of the tissues of the heart, skin, blood vessels, tendons, and other body parts, and it weakens the tissue structure, especially in the aorta. And the man in the picture is Harris Charalambous, and he was a promising college basketball player that didn't know he had Marfan syndrome. And he died at the age of 21 in 2006 during practice when his aorta failed and burst. So people with Marfan syndrome generally go undiagnosed unless they have an incident like this one because they don't know they have any obvious symptoms. So traits can appear in 2 or 3 forms, or they can occur in a range of small differences with a continuous variation. So the more genes and environmental factors that affect a trait, the more continuous the variation. So for instance, human height and eye color are 2 traits that vary continuously. So they have a lot of factors that influence what phenotype you actually see. So the chart shows continuous variation in height among male biology students at the University of Florida. And so if you look on this left-hand side, that's what that shows. So the students were divided into categories of 1-inch increments in height and counted, and a graph of the resulting data produces a bell-shaped curve, which is an indication of continuous variation. So the picture on the right shows the spectrum of eye colors that humans have from dark to light blue. So environmental factors can also affect gene expression, and this in turn affects the phenotype. So this can include seasonal changes in animal fur colors. So the color of the snowshoe's hair fur varies by season, that you can see in the pictures. So in the summer, the hare's fur is brown. In the winter, it turns white. And so this gives the appropriate camouflage of the fur against predators. So other environmental factors phenotype include spine growth in the presence of predators and daphnia. And these are water fleas. So these organisms develop a longer tail spine and pointy head as defenses in response to chemicals emitted by the predatory insect. And also plants, including the yarrow plant, have been shown to grow to different heights when grown at various altitudes. So as we saw in the last few slides, not all traits appear in Mendelian inheritance patterns. So an allele can be partly dominant over a non-identical partner, or it can be co-dominant with it. And also multiple genes can influence a trait, or some genes can influence many traits. And finally, even the environment can influence the gene expression through proteins being expressed in response to temperature or chemical changes in the environment. Any questions on this chapter? So that's all I have for tonight. I'll stay on for a couple minutes if you do have questions. Otherwise, I will see you on Thursday.